This recorded session is dedicated to the last group of medically significant bacteria that we will cover for Module 4. So we'll talk about two species of non-spore forming bacillus. We will cover Corynebacterium diphtheriae and Listeria monocytogenes. So let's start with Corynebacterium. So C. diphtheriae is the agent of the disease diphtheria. Diphtheria has uh, been associated with upper respiratory tract infection, so particularly inflammation of pharyngeal tissue, so pharynx. No? So pharyngitis is the um, essential manifestation of C. diphtheriae infection, and um, as a key example of non-spore forming bacillus pathogen, bacterial pathogen. So C. diphtheriae is known to is actually um, um, its infection is actually preventable. So in our contemporary time, diphtheria is uh, not common anymore. Okay, so this infection is actually vaccine preventable. Okay, um, but in at-risk population, especially the non-immunized ones, the disease may still cause significant clinical effects or clinical manifestations. Okay. So for the morphology of the bacterial pathogen, so C. diphtheriae is non-motile. It's non-encapsulated and could be an aerobic or facultatively anaerobic microbe. No? There are actually several biotypes of C. diphtheriae, which I think I did not reflect or it's not reflected in this handout. But there are different biotypes beyond or below under the species C. diphtheriae. So there are C or Corynebacterium gravis, Metis intermedius, and Belfanti. So C. Metis is the causative agent for mild diphtheria. C. intermedius for the intermediate type or the intermediate form, and gravis is the more or the most severe. Is a causative agent for the most severe type of diphtheria. Okay. In terms of the appearance, so the cells look like pleomorphic or appear pleomorphic. They can be irregularly shaped at times, may form V or Y shaped figures with club shaped ends. Okay, so here you can see the stain a photomicrograph of Corynebacterium diphtheriae cells. Okay. The transmission of this bacterial pathogen is primarily via the respiratory route, by respiratory droplets from a carrier, from a host that allowed the reproduction or where the reproduction of the species has been extensive. Okay. As a ubiquitous organism, there are several reservoirs of the species can be universally found in soil, in water, in plants, okay, in animals, as well as in humans. No, actually, humans are the most extensive hosts of the group of this bacterial species. Okay. Now, I remember in our on-site lecture, I talked about the diphtheria toxin as an example of the toxicity factors that bacterial pathogens can can utilize in order for them to be more virulent and more invasive, or more virulent, invasive, and essentially severe in terms of the in terms of the pathophysiology of the virus to a particular human host. So as I mentioned, it's a primary cause of pharyngitis, so it colonizes the pharynx. The pharynx is the tissue of choice for this bacterial species, ensuring or as it progresses to cause um, severe disease. It can form grayish pseudomembrane, primarily made of fibrin, leukocytes, dead or necrotic epithelial cells, and the cells of the bacterial species itself. And those pseudomembranes can block the passage of air to the lungs. Actually, the pseudomembranes has something to do with the species epithet of the bacterium. So diphtheria means it's a term associated with leather or hide, because the pseudomembranes that are produced by this bacterium along the airways actually has a leather-like appearance. Okay, so here you can see a patient with pseudomembrane of associated with seed with Corynebacterium diphtheriae. Okay. So as a review, I talked about last time. I talked about the AB toxin, which is a powerful exotoxin released by or produced by Corynebacterium diphtheriae. So this AB diphtheria toxin, as a review, okay, is composed of an enzymatic component, enzymatic unit, the A unit, and a receptor related unit, the B unit. No? So the B unit has a particular receptor in the host the host cell membrane, while the A unit is the enzymatic one. No? It's an enzymatic component. So the B component, it binds to its particular receptor on the surface of the cell membrane, allowing membrane lipid to be cleaved or to be uh, to lose its integrity, hence allowing for the A unit to be translocated inside the host cell. And the A unit interferes with protein synthesis. So it makes the elongation factor. It makes elongation factor, which is an important molecule during protein synthesis, to be non-functional. So there is stoppage of protein synthesis associated with diphtheria toxin. And clinically, that can result to heart damage, damage to neural cells, and nerves may be affected. The diphtheria toxin or diphtheria intoxication or toxicity can lead to partial paralysis of the host. Okay. May also cause inflammation of the myocardium of the heart, and in certain cases, okay, peripheral nerve palsies. So there's neural involvement if neural cells are affected by the toxin. So the manifestation is similar to the Guillain-Barré syndrome. Okay. So how is C. diphtheriae treated? So it can be treated with the use of an antitoxin. Um, for antibiotics, there is penicillin and erythromycin, which can be used. And there is also a vaccine. As I mentioned, diphtheria toxoid has been utilized as a universally accepted vaccine for this bacterial pathogen. Okay. What else? Okay. Now, so I think more on Corynebacterium, um, which I did not, not what is not reflect or are not reflected in this handout. So, C. diphtheriae, the infection, the disease cause, actually is very similar to other disease conditions. And the clinical manifestation of C. diphtheriae is similar to pharyngitis that's caused by a virus. Can also pharyngitis that's also caused by streptococcus, or an oral cavity infection caused by yeast like candida. Okay, but it's important, the definitive diagnosis of the bacterium is very important to be able to properly treat the disease or the diseases that are caused by it. Okay, now let's have a look on the last medically significant non-spore forming bacillus. So let's have Listeria monocytogenes. So Listeria L monocytogenes causes the infection listeriosis. Okay, so as a facultative intracellular bacterial pathogen. So it's um, taking advantage of the immune cells hunting it down in order for it to survive and become more invasive or become more distributed in the human host. No, the name was derived from monocytes, which are proliferating during infection with this bacterial pathogen. They appear as slender short rods, can appear in short chains as diplobacilli. Okay, so here you can see two rod shape or two bacilli shaped cells that appear in chains. No? Okay. When in a liquid medium, they can be observed to have or they can be observed to manifest through motility, and it is known that L. monocytogenes is the only gram-positive organism which has an associated endotoxin, a structural toxin. Okay, so the ingestion of contaminated dairy products is associated with the infection uh, by this bacterial species. The thing with L. monocytogenes is it is actually capable of thriving in cold temperatures, in very low temperatures, even in refrigerator temperatures. They can grow as in ambient as low as 2.5 degrees Celsius. Okay. There are several virulence factors associated with L. monocytogenes. There are a few that are not included here, but the ones that we have included as shown are the flagellar antigens, since flagella allows it to be motile, to um, to be capable of adhering more on the host cells, like the enterocytes that they target if they're consumed as primarily they're consumed as foodborne pathogens. And they're also able to produce hemolysins, no? Which are cytolytics. Additional virulence factors for L monocytogenes include several more what they call this several more molecules. So we have surface proteins called internalins, which allow them to attach to the host cell. There's also an because as facultative pathogens, so they are phagocytosed by immune cells. And when they are phagocytosed by immune cells, they are encapsulated in a vacuole. They are within vacuoles of the host cells that phagocytose them. So in order to escape vacuoles, they secrete, they express lysins, so listeria lysine O, LLO, which allows them to escape the host cell vacuole. Within the host cell, they take advantage of moving between cells via actin polymerization. So they are able to, they have associated proteins that can assist with actin polymerization so that they can move from one cell to another. More so, they also express phospholipase C, that will help them not only escape the cell vacuole, but also disrupt the cell membrane of the host cell that they are going to or coming from. Okay. Now, clinically, listeriosis exhibits tropism or preference for nervous tissue, and the species is a common cause of meningitis in at-risk populations such as newborns and immunocompromised, immunosuppressed individuals, the pregnant women, the elderly, and also those with immunodeficiency sicknesses. Okay. For the case of pregnant women, they prefer the placenta. They can actually cause spontaneous abortion or stillbirth. Okay. And in cases when the newborn that has been infected by this species survives, some manifestations may still appear, such as meningitis and widespread distribution of the pathogen in the blood. No? So I've mentioned all of these already. Pregnant women are at risk, no? Especially that as a foodborne pathogen, L. monocytogenes can, uh, L. monocytogenes can, it's actually very hard to control. So it's a very tricky foodborne pathogen to eliminate since it is able to, what do you call this, it's able to reproduce itself in freezing temperatures. So, sterilization may not always work all the time. But the thing is, L. monocytogenes is only associated with improper food handling practices and conditions. So with proper food hygiene, definitely it is a preventable infectious agent, or it is an infectious agent that can be confined, or that can be the risk of it transferring from its source, from the food to a potential host, is actually lower, no, or it can be lowered. Okay. So I mentioned this already. So the perinatal transfer, or the perinatal associated infection, perinatal associated listeriosis, granulomatosis infantseptica. So there is transmission of the pathogen from the mother to the child, the neonate, usually via intrauterine route during delivery of the child, delivery of the neonate. Okay. How is L. monocytogenes treated? So there is effective set of antibiotics that can be used, ampicillin and trimethoprim sulfamethoxazole. Trimethoprim binds to dihydrofolate reductase and and inhibits the reduction of DHF to THF. That will inhibit DNA synthesis in the bacteria, in the species. And S-methoxazole counteracts fatty acid synthesis. Okay. I think this is the last slide. So we already covered all of the medically significant bacterial pathogens to be included in this module.